These are real images of cancer cells changing shape. They do this in order to metastasize and invade new areas of the body. This change of shape has been observed for many years, but only recently this has been a real research focus. By understanding how cancer cells change shape, could we finally control metastasis?
Cancer becomes a lot more difficult to treat when it spreads to other parts of the body. This is known as metastasis. In fact, metastasis is responsible for around 90% of mortality from cancer. So today I hope to understand the relationship between cell shape and metastasis, and that's why I'm here today at the Institute of Cancer Research in London to speak with experts in their field and to see what they're doing about this dilemma. It's off these corridors that scientists are working on some of the most groundbreaking cancer research in the world. The ICR has 100 teams dedicated to specific areas of cancer. Today I'm looking for the Dynamical Cell Systems team, where their research focuses on how cancer cells change shape during metastasis.
The team is led by Dr. Chris Bakal, who told me a bit about some of the topics covered in his lab.
We study cell shape because cancer cells, when they spread throughout the body in a process known as metastasis, absolutely have to change their shape in order to leave the primary tumor, get into the bloodstream, get out of the bloodstream, and then find a distant site in the body. They absolutely have to change their shape. So we think that if we can understand these shape changes and block them, we can prevent metastasis from occurring. And because 90% of patient deaths are due to metastasis, are due to the process of cancer cells changing their shape, we think we can make a major impact on cancer mortality by blocking these shape changes.
And is there anything currently available that kind of stops metastasis or targets it?
It's a really good question. In fact, there's not that many anti-metastatics available. Most of the therapies that we have right now target the primary tumor and target their survival. So it would really be a new way to treat cancer by targeting the metastatic process.
So what kind of methods do you use to investigate this?
So to study cell shape in the lab, we use systems biology approaches, we call them, where we take lots of images of lots of cells, millions and millions of cell images per day, and then we use artificial intelligence methods to analyze those images, and then computational and statistical methods to analyze the data that comes from those images. A lot of the techniques we use are similar to the AI-based approaches that people use to analyze images on their phone, or for example, they use on CCTV cameras in the tube. It's the same kind of methodology we use. We use computers to analyze the images, and then once we have that data from those images, we use statistical methods to analyze that data further.
What specific projects are going on in your lab at the moment?
So one project which I'm really excited about is a project work that's done by Patricia Pascual Vargas, a Stand Up to Cancer funded PhD student in the lab, who's identified a new gene that we think is absolutely critical for breast cancer metastasis. So she's trying to understand the role of this gene, but also trying to find a way to block it in metastatic breast cancer cells. In fact, we can go talk to her right now if you want.
Oh, great.
For the last 3 years, Patricia has been studying a particular family of enzymes. I sat down with her to understand what they are and their significance in cancer metastasis.
So how is cell shape regulated in normal eukaryotic cells?
Cell shape in normal cells is regulated by rho-GTPases, which basically control all of cell shape. And in cancer, this is basically goes wrong, so they can become overactive and confer cancer cells with the ability to basically change shape and be able to spread and metastasize around the body.
Why is this significant to triple-negative breast cancer?
So triple-negative breast cancer lacks 3 receptors, which makes it very difficult to treat, and it's a very metastatic cancer with high rates of relapse. So we need to find more targeted therapies to treat it. And within triple-negative breast cancer, it's been shown that there's an upregulation of the expression of rho-GTPases. So we think that this is what's conferring their ability to be more metastatic. So they're changing their shape more, and this is what's allowing them to spread more. So if we can pinpoint which genes are regulating these changes, then we can stop the cells from changing shape and so prevent metastasis from occurring.
And what methods do you use to investigate this?
So first of all, what we did was do high-throughput screening with Si Rnas. And so we did it on two triple-negative breast cancer cell lines. We used the Opera high-content imaging system, and basically we knock these genes down, we stain them for cell shape cytoskeletal stains, and then we upload all of this to a software which we then can extract features on changes in cell shape, essentially, even like nuclear roundness, cell shape roundness, etc.
And what stains do you use to stain the cells?
So we stain for tubulin and actin, which basically gives information on how depleting these genes is changing the cell shape of the cells. And then we also stain for the nucleus or DNA.
So is this something that we can visualize in real life?
Yes, so we do two types of imaging. We do fixed cell imaging, where the cells are static and dead, and we're just quantifying cell shape. And we also do live imaging on total internal reflectance microscope, which basically allows us to see how the cell is interacting with its microenvironment and how it's moving around, and how cell shape is affected. If you like, there's some videos I could show you of these cells.
Yeah, that'd be great. So what type of cells are these?
So these are triple-negative breast cancer cells derived from MDA and B231s.
And what does the red show?
So the red is showing focal adhesions, which is where the cell is making contact with the matrix, and it shows us the direction of where the cells are going. So for example, this one is just a triple-negative breast cancer cell. We've done nothing to it, and the adhesions are at the edge of the cell where they should be, and the cell is moving in the particular direction. Whereas if we look at this cell, it's lacking the gene that we are studying right now, and you can see that the adhesions are much more dynamic, and they're all over the place. They're not really like at the edge of the cell, and it's not moving in a particular direction.
That's really fascinating. So what is the therapeutic potential of this?
So if we can inhibit this gene, then we can prevent the cells from changing shape and thus prevent metastasis. And so there's an inhibitor that's already been developed for it, so we know it can be inhibited. And there's also in Sutton, the ICR can develop drugs, and they would take the next steps.
So what are the next steps for this research?
So currently we're trying to understand what the mechanism is, so how this gene is regulating cell shape and driving invasion in triple-negative breast cancer. And then we would try to test the inhibitor in vivo in mice and see if it really is decreasing metastasis.
And at which stage during disease progression might this be useful?
So we think that it'd be good to give the inhibitor to patients after tumour resection, so then in this way we could prevent metastasis and then prolong their lives.
Patricia's is just one of hundreds of projects here at the ICR that is looking at ways in which we can help reduce cancer patient mortality. With research like this, we are one step closer to shaping a promising outlook for the future of cancer metastasis.